Thank you for inviting me. I'm really privileged to giving you a talk entitled as Making Neural Networks Exhibit Robustness Under Adversarial Conditions. This is a joint work with General Electrics, and I was with MIT, in the past 2 years, and we had the privilege to be found supported by the intelligence agency, who were interested in recognizing human faces, human body, posture, gesture, the gait movements, for instance, to recognize people, actually, identification of people from a distance. And under turbulence, for example, and other perturbations you might think of. But I'll formulate my talk. I'll actually divide my talk into two phases. I'll first tackle two questions and hopefully give you some of the findings that we, as a lab, that we have reached during my time when I was at MIT. So let me give you a little bit of background. I got my PhD back in 2012, so 12 years ago. I was a researcher in different companies. I re-entered academia and Met University until 2022, and then re-entered again this year. When I was away, I was at MIT, but as a visiting researcher. And then I started as a professor in Boğaziçi University this year. I got a couple of visiting positions, and I'm interested in information coding theory, distributed systems, signal processing, telecommunication technologies, optimizations, and more recently, neuroscience and its impact on and human vision and its impact on the artificial intelligence model development, training, and whatnot. I've got also interesting collaborations spanning from some of the local universities to CTTC Spain and some of the European research centers. And also a couple of universities. So let me just briefly give you what this talk is about. I'll be focusing on modern, so if you already know that modern face recognition systems, they seem to exceed the humans by a large extent. And algorithms that see better than humans, now we are basically talking about those algorithms and their long-term effect on the human social system. However, we really want to actually ask the following question, because we're basically kind of curious that if this claim is rightfully true statement under different adversarial conditions, particularly conditions in which the neural networks are known to not generalize very well. So we'll be focusing on those conditions in this talk. And as I said before, IRP is interested in identification of humans from a certain distance. And as you can see, that if the objects that you're seeing in this picture, they're all humans and therefore on the same type. And that actually increases the challenge of identifying a specific person in this picture. However, pictures like that, if the type differs, now the objects are different. Now it's kind of easier now to detect a human being, for example. So this challenge need to be addressed through different mechanisms, and the mechanisms in which we find interesting solutions is the human body, the human vision, and the brain mechanisms. So that's why we're bringing in a neuroscience perspective to this problem by having this vicious cycle. We'd like to first of all ask the following fundamental question: do humans perform better than the machines? If they don't perform better than machines, then we have to try ad hoc approaches to improve machine vision systems. Okay, so we don't really have anything to get ourselves inspired from the human body, human brain. However, if your answer to that question is yes, like do humans perform better than machines, if it's a yes, then we start understanding how humans achieve their superior performance by running experiments, for instance. We'd like to translate those insights from human vision to machine vision and test machine vision systems and identify their vulnerabilities. And finally, we test humans in these settings, and then we compare the machines and humans, see if that the machines are benefiting from the mechanisms we identify through experimentation, with humans. Or maybe some other biological systems, as macaques, for example, or monkeys. That's the basic idea behind the neuroscience perspective. So here is there are three different legs into this project. First of all, and I will talk about today, is physics. We'd like to, for example, make sure that the humans and machines are comparable. Most of the time, humans and machines, when they're compared, the setting in which these two mechanisms, the intelligences are compared, is not apples to apples in many cases. So therefore, we would like to develop a technique. For example, we would like to analyze the human front-end optics of humans, and then try to figure out that if the machines could use the same type of optics, front-end optics. So therefore, whatever the optics basically leave you as important data or information, then the same information will be processed by the visual cortex of humans and the machines, such as AlexNet, ResNet, all these computational techniques, which are to be mimicking the human visual cortex, for example. Okay, so in the same project, we looked into different psychophysics, such as eye tracking, neural dynamics through EEG, MEG. We also looked at computational mechanisms to see if they're generalized, once you're inspired by the human mechanisms, and translate such mechanisms to the machine development or model development, then see if that helps you. And we have got a bunch of other little projects within that, so I'd like to focus on two. So first of all, let's start with this specific online study. In this study, you are shown a face of an individual, but those faces could be, turned around, yaw and pitch changing, for example, or the viewing distance also could change. For example, the one on the right is taken from 500 meters, if I'm not mistaken. And as you can see that it becomes blurry, and because of the turbulence, there's a motion blur as well, and it's kind of dynamic, it changes over time. And you're asked to choose that individual from the 5 gallery images. So the query image is from a specific data set that has got all these variations, and the gallery images are from, these people are all front face images, and you're asked to choose which identity is the one that you're seeing in the query image. Okay, and if you look at the results, so what we did, we basically run some experiments on humans, and we also run some convolutional neural networks, which are pretty known in the literature, the ones that are on the left. And the performance you're observing right now is the average of these models, and also the average of the human participants in our experiment. And as you can see, so there are 7 different variations of yaw and pitch, and the definitions of yaw and pitch are shown on the left. You can see the face could be looking up, could be looking down, or could be looking right or left. So you only can see the cross section. The results are kind of interesting because, as you can see, that if the images are only 2 meters away, then the results, the accuracy performance of the CNNs are kind of close to the human performance. However, if you change the yaw and pitch, or if you change the distance, like for example 1,000 meters, 1 kilometer, then the performance degrades both for humans and the machines. However, the degradation for humans is less sharp than the machines. The machines kind of suffer a lot due to these adversarial changes. So the first question we would like to ask because the comparison here was a target for many audience when we give this talk. They always criticize, you're not really comparing apples to apples. You should be comparing them, making sure that they kind of have access to the same information. So therefore, then we start, coming up with specific questions that would address such a concern in the audience. So we've asked the following question: how well do the humans recognize face as a function of viewing distance? And there have been some studies, but the idea here is to see if run some experiments and see if humans can identify a specific individual at a given distance and see the accuracy of that identification performance. And this is from this paper. As you can see, that they also managed to see if the internal features and external features of the face could play an important role in the recognition performance. But what we're trying to show with this particular plot is that if you just look at the internal features and also the external features, which are green and the magenta, not magenta, but cyan. So they show, something you expected, that as you get closer, then you expect better performance, and as you get far away from the optotype, from the individual, then you would expect less of a performance. However, if you take the union of the external and internal features, you get something, the dotted line, the dotted black line. Okay, so that's the union of the accuracy performance. But if you keep the whole head as is, without changing or without dissecting into internal. Oh, merhaba hocam. Tamam. Okay. So the interesting takeaway from this picture is that the red line is the whole head without dissecting internal and external features, whereas the dotted black line is the union of these internal and external feature performances. So therefore, what we get from this picture is that the human brain is processing faces using holistic mechanisms. So it has to be a whole. So the configuration of the eye to the nose and to the ear and etc., they have to actually comply a certain specific set of distances, which makes the holistic processing work. Okay? So that is, I think, one of the interesting takeaways from this picture. Okay, so as I said, evaluation problem. How can we use these results as a benchmark for evaluating the performance of a computer vision system? So there are two important real-world properties that current state-of-the-art computational models are missing. First one is time and space. And you're familiar with the space-time concept from Einstein's relativity. Now I will try to adopt that kind of idea to the neural networks, because there is no time defined for neural networks. Most of the modern computational neural network mechanisms do not have any time. You basically apply an image and then get an output. And there is no space. Space concept is also absent in the definition, original definition of the neural networks. And of course, we need to translate to higher level descriptions of these concepts, because these do not make sense in the context of designing a neural network model. Okay, so we need to understand how these two concepts, important concepts of 3D geometry that we're living in, could translate to a neural network model. Okay, so let me just skip those because we already have done. So I would like to just come to this experiment. So first of all, we would like to change the front end. Okay, the way that we change the front end, we first run an experiment, and the experiment is as follows. We've got 20 participants, and they are all normal or corrected to normal acuity. There are 5 queries for each of 15 resolutions and at 16 different distances. And we have got this 4x4 image, you can see, and one of them is blurred with a specific Gaussian sigma, okay? And then we ask the participants to find that odd picture, odd face, okay, from the rest of the others. Okay, and then we basically run this experiment for different distances and then ask them this question. And based on the accuracy of the answers, we finally understand, because as you know, that our eyes keeps doing lots of low pass filtering. As the optotype, the individual or an object, is getting away from our eyes, it gets more blurry and blurry, okay, because of the way that our eyes work. So you can see this in this picture. You can start from the right end and then move closer and closer to the eye. At some point, you will get the following: the low resolution image, that's the odd one, the odd one. The odd one's resolution is going to meet the high resolution image, which is the rest of the other three, at specific points. Okay, and that's what we are looking for. We would like to understand where exactly the image resolutions become the same, although they are different. But at a specific distance, they look the same to a human eye. Okay, and that distance is going to give us that specific resolution that is available to the human eye. Okay. So let me just skip that detail. Let me get to this. Although it looks a bit complicated, but the takeaway is very simple. The y-axis now, instead of using trigonometry and try to figure out, for example, the angle of resolution, angular resolution, what we actually translated everything into something called effective resolution. Effective resolution is the number of pixels that are available to the human eye at a given distance. As I said before, as the distance increases, then the resolution decreases. Okay? And that's exactly what we found. The blue, yellow squares with the dotted blue line, it's the data. It's the experiment results. And we also had a model, a simple theoretical model, that kind of overlaps with the data almost, might be not to close distances, but definitely for longer distances. And you can see that the resolution, effective resolution, for a fovea, the fovea is basically the highest resolution part of the eye, at the retina, and then there's also parafoveal region, which basically is the lower resolution region of the retina. So this line that you see that I called parafoveal region and foveal region, as you can see on the bottom figure, is those two specific regions of the retina. Okay, and the fovea has lots of photoreceptors in it, and most of the literature assumes these distribution of the photoreceptors to be uniform. However, parafoveal region actually has a drop in the photoreceptors, particularly cones, okay, which are responsible of seeing color and light vision. Okay, so you can see that we can now use this kind of functional relationship between distance and effective resolution to train, for example, neural networks, because all we're looking for, this functional relationship. Maybe let me just jump all these, and let's come to the takeaways of this simple study. First of all, we realized that particularly for the short range, we have a disparity that still persists between our predictive model, the theoretical model I was referring to earlier, and the experimental data. And differences in the errors made by the subjects, they're also not really congruent. In other words, they cannot be just explained by their age or acuity, contrast sensitivity. Okay, there's something also going on at the back end of the visual system, which should actually explain this inconsistency in the way that the people are making errors. And having a common human visual front end, I think we should be able to compare the humans and neural networks, which I'll show you in a simple example, how I can do that. And the other is be able to understand the human brain mechanisms with the available input resolution. Because it turns out that as the resolution drops, the human mechanism for recognizing human faces kind of changes. Okay, so we basically have, although implicitly we don't even know, we actually use different techniques when we recognize someone. For example, if that someone is someone that you're familiar with, like a friend, you can actually easily recognize that person from 1 kilometer away. But if that someone is not someone you already have seen or have talked to, then that becomes hugely challenging, to be able to know that person. And this person has to come closer, for that recognition to take place. Anyway, so let me just show you one specific example, what I mean by be able to compare humans and neural networks on the same grounds. Well, here's a simple training of a CNN, a performance of a CNN with the human front end. So I made sure that the human front end is used in this inference experiment as a function of physical distance. So now we can talk about the network performances, okay? Rather than, for example, simple noise parameter or some other change, for example, in your experiment, now you can actually change physical distance, which is three-dimensional property of our real world. And you can actually now show top 1 accuracy of these CNNs and compare them as a function of distance using this front end. The other is, of course, make sure that we were able to understand the human brain mechanisms with the available input resolution. And for that, we run some experiments, psychophysical experiments, most prominently the electroencephalography, EEG experiments. Okay, let me just jump to that as my second phase of my, my talk. And please interrupt me if you've got any questions. So let me just start with the brain network modularity, okay? This is defined to be the degree to which functional brain networks are divided into subnetworks. Modularity is assumed to be beneficial to develop a robust brain, okay? So you can actually look at the human brain and see that the specific regions of the brain, like visual cortex, is responsible of seeing things. And there are also parts in the temporal cortex, for example, responsible of hearing things. And the frontal cortex, for example, responsible of making calculations. Mathematical, for example, developments are due to that specific region of the brain. So that modularity seemed to be the main reason for robust recognition in the brain. Modularity does not typically form in computational models, by the way. With standard training and architectural design like backprop, for instance, or layered design of such we see in AlexNet or ResNet, for example, or skip connections, for instance, it doesn't form automatically. Modularity can be enforced through training or optimization mechanisms. So first, for example, what are these examples of optimization techniques or learning schemes? For example, biomimetic training regimen. What does that mean? Well, if you just look back here that I'm showing you at the bottom of the slide, what you're seeing is the first 6 months of a baby and how its vision matures over time. And the far left is maybe perhaps the first week or two. You see it's all black, white, and it's all blurry. And as you move forward in time, you see at the end of the 6 month, by the end of the 6 month, you start seeing a clear picture with all colors, okay? So this change, if you apply the same change to neural network training, you get a lot of performance improvement. And that also leads to a bit of a specific modularities that emerge in your neural network training. So that I find very interesting because we're basically looking at the human development and then copy something from human development or mimic something that you see in the human development on a neural network training, which basically is the kind of the same model that we believe that resembles to the brain networks. And if you basically make sure that the baby humans, they exposed to a certain specific environment and make sure that that environment is around the same environment that neural networks learn things or objects or their relationships, then we see a performance improvement. Okay, so what my objective was as part of this idea is to look other mechanisms which exist in the human brain and see if we can apply the same mechanism to the neural network training or neural network architecture design. And that specific mechanism I realized is the spatial frequency processing, which is something that people alluded to in the literature. But I just looked into it because I wanted to see if I can actually do repeat their results, if I can see the same results that they are reaching in their experiments, and also come up with a mathematical model that will explain this spatial frequency processing, both in humans and as well as in the neural networks. So the hypothesis in the literature is coarse-defined processing. In other words, it proposes that the coarse information carried by the spatial frequencies of the visual input guides the integration of the finer high spatial frequency details. In other words, when we see a face or a body or any image, a natural image, what we basically do in the brain, the low spatial frequencies are processed earlier, and then they reach up to the frontal cortex. And then the high frequencies, high spatial frequencies are processed later in the visual cortex, and then they kind of meet in the middle, and then the low spatial frequency guides the high spatial frequency, to complete the perception, okay? So that's what most of the people that run psychophysical experiments reach up to when they completed their analysis or their research. They come up with this kind of, more or less the same conclusion. And you can also see the same course-defined processing in other biological mechanisms, such as macaques. And instead of running, for example, non-invasive techniques like EEG, MEG, you can also run invasive techniques like neuron-level signals. If you collect those signals and look into them, you will realize that there's a difference in the temporal direction. There's a difference between the low and high spatial frequency processing. Difference in the delay between the two. Okay, well, spatial frequency processing in the brain. So let's think about the time and order. So as I said before and earlier, there are two dimensions that I wanted to look into, time and the space. The time component doesn't apply to a neural network. Okay, there's no time defined. Even like humans, for example, time is relative. So we don't really having hard time understanding the concept of time anyway. So in neural networks, we will consider order rather than time. So concept of order in which events occur is more basic than the concept of time. So therefore, I'm going to look into the order of low and high spatial frequency processing in neural networks. So maybe the most straightforward idea is to have different networks that would process low and high spatial frequencies, and then maybe a specific merging mechanism that can actually merge the two, and then make a final classification. Okay, something like what you see in the bottom of the slide. Okay, to make sure that the low spatial frequencies are guiding the high spatial frequencies, perhaps you have to first process low spatial frequency, that's the order part, and then make sure that the high spatial frequency is benefiting from the low spatial frequency, classification or a task completion performance. Okay, so the way that I would like to compare, if there is a difference in different neural network architectures, see if there's a difference between processing high and low spatial frequency. So what I did, I looked into the activation maps of a simple AlexNet. I'm sorry, not AlexNet, VGG16 this time. I also have other networks that I've run earlier. I put the low spatial frequency and high spatial frequency as input, and also the clear picture. And then what I did, I compared the activation maps of low spatial frequency activation maps to the clear picture, and also the high spatial frequency activation maps to that of clear picture. Okay? And here are the results. So what I did, the comparison is done based on SSA-ADM score, which is an image comparison, a subjective metric. And you see that the different convolution layers, you see from the left to right, is the different convolutional layers. And the y-axis is the similarity of the activation maps. The low-pass filtered images seem to show a more resemblance to the activation maps of clear pictures in the earlier layers than the later layers. And for the high-pass filtered images, I see kind of the same thing, but a reverse direction. In other words, in the earlier layers, we seem to see some similarity, but that similarity seems to increase a little bit, but still increases for high spatial. So this kind of gives an idea that low spatial frequencies might be preferred by the neural network earlier in the processing, in the earlier layers than later layers. And high spatial frequencies might be processed later in the layers rather than earlier. So therefore, this kind of in line with our hypotheses that the low spatial frequencies might be guiding the high spatial frequencies. But this is just a simple network. How about other networks? For example, we know that ResNet architecture is from the most influential models right now in the literature. They have got these skip connections, and the basic idea behind skip connections was to help the optimization, right? Because the optimization wasn't really converging very well, and therefore they come up with this idea of skipping the connections, and then finally they get it done. However, that's not necessarily inspired from a human processing, idea of processing in the human brain. So I also looked into ResNet. see if there's a kind of a similar observation. But unfortunately, I failed to see that. You can see that these pictures, the same picture, the y-axis is the activation maps, similarity, and x-axis is the different layers in the ResNet. And I failed to see the kind of the same trend. Perhaps it's a deeper network, perhaps some other reason. And if you look at the performance of these networks, particularly VGG16 and ResNet 50, the VGG16 seem to work better than the ResNet 50. ResNet 50 doesn't have that property. In other words, low spatial frequencies are processed earlier, and high spatial frequencies are kind of guiding the low spatial frequency results. But VGG16 seem to have that property, okay? And if you look at those results, one thing that you realize that, and this is another example, that architectural dependency is obvious. So there's a difference between the architecture of these networks and their processing low and high spatial frequencies is different. Perhaps human robustness is encoded innately into the substrate. In other words, the hardware structure, rather than just the training, okay? Spatial frequency preference of the network. So let's look into that. Explicit tracking schemes needed deep down the network, okay? So the conventional schemes of training a neural network might not be the best solution. How about training regimens that could benefit learning? Well, initial low spatial and late high spatial frequency exposure may help you with the overall training of the neural networks. So how does the external stimulation shape architecture, and how do the dynamics of architecture change the requirements of training? So these two other fundamental questions are important, in training a neural network, because as you can see that if we haven't changed the training yet, but if you change the training or if you change the architecture, then the requirements of training and also model development could change depending on, because these are coupled tasks. You can't really separate the two. And here's the question: how does human visual system handle the spatial frequency decoupling? Because that needs to be decoupled somehow. And we realize that there are two different pathways in the brain, magno and parvocellular pathways. And the visual system functionates the processing of low frequency and high frequency information into these two pathways. Therefore, there are separate hardware in the brain. Okay, that's something that we realize through psychophysical experiments and some of the invasive techniques. And processing of high spatial frequency is delayed and guided by the low spatial frequency for final perception in the infratemporal cortex and up. So this observation, of course, depends on the exposure time. If it is smaller, for example, 100 milliseconds versus 500 milliseconds, there's a difference between the two. And the demands of the task and the visual characteristics of the stimuli. Okay, so I'm going to come back to the ResNet because I haven't seen this low and high spatial frequency, decoupling. So therefore, I started studying ResNet a bit more, and then I realized that there are already some work that kind of says that ResNet is a model that kind of mimics the human brain. For example, a recurrent neural network, which as we know, the brain is a recurrent neural network, and this can be used to generate the basic residual network if input remains the same with respect to time. So these are kind of interesting findings because what they're suggesting is that the ResNets could be a really good biologically plausible model. Okay, so I actually looked into that and said, okay, I'm going to use ResNet. Why not? And I haven't seen the low and spatial frequency, separation. So let's get started. Let's just introduce this time lag frequency decoupled networks. What does that mean? Well, this is the ResNet 50 you see on the right. And what I'm going to do, I'm going to actually feed forward, as you can see, for example, this DS. Don't worry about the details. The DS is one specific block called downsampling block, and R's are the residual blocks. So what I'm doing is the output of the DS, okay, without going through the filters of the next block, they are kind of feed forwarded. But what I feed forward is the high spatial frequency rather than the low spatial frequency. So the DS block is modified in the following way. You see that this filter, the yellow one is the basic building block of a ResNet downsampling block. But the other two are, you see that we actually the data input going through a filter called Gaussian filter, let's say, low pass filter. And then we divide this frequency spectrum into two, low and high spatial frequencies, and they go through different filters, okay? And the high spatial frequency is feed-forwarded to the next cell, okay? Or in this case, is R cell or R block. Okay, here is details. So there are three basic components here. One, frequency decoupling. We decouple the frequency spectrum into two, called low and high spatial frequencies. The second one, time lag processing. In other words, we feed forward the high spatial frequency to the next cell. And finally, the fusion. The fusion are done using these weights, but those are subject to optimization again. So we don't know, whether or not the low spatial frequency is important and high spatial. We just let the network learn that, okay, and see if that's going to give us something, provide some insight. Okay, and one note that we have low-pass filtering that can act like an anti-aliasing filter. For those of you who are with a signal processing background will know that anti-aliasing filtering could help with distortion. And low spatial frequency and high spatial frequency are processed concurrently or with delay. Okay, and this delay is, as I said, based on order by using these extra connections. Skip connections in this case, but only feed forwarding high or low spatial frequencies. So the idea is to introduce additional paths and extra weights of fusion, and training not only optimizes the parameters of filters, but also the weights of fusion, and that would cause a pruning of the network, because if one of the weights is 0, then that's actually corresponds to pruning of the neural network in this case. An idea can be generalized, apparently. This is only for two different, dissection. The spectrum is divided into two, but you can actually divide the spectrum to more regions, using this kind of idea. Okay, so I'm going to be focusing on, so here there are two parameters called the breadth of the network and also the depth of the network. I'll be simplifying my presentation. So the breadth of the network will be always 2, for example. So I'm going to have a lower high spatial frequency. But the boundary between the two will be optimized. So we don't know where that boundary is. And the depth of the network, of course, depends on how much lag you want to introduce to the model. And I'm going to probably show you a couple of different lags and see if they make sense to you. Okay, so here, what I'm analyzing here, innate characteristics gets encoded into architecture. And here, one manifestation of this could be learning from few examples. As you know, in the literature, one-shot learning, few-shot learning. So what I did here, I just changed the test ratio. I started from the 95% test ratio on the far left, and on the far right, 95% of test ratio, which means only 5% of the data set is used for training. And as you would expect, the performance will be impacted by this. However, if you look at the yellow ones are the biologically inspired model, that's called ResNet Bio. Their generalization performance is better. But not only that, the performance gap between the conventional scheme and the biologically inspired version is getting bigger and bigger, in terms of percentage, as you go from left to right. Okay, so that's, I think, one of the with CIFAR 100. By the way, data set CIFAR 100. So it's, I think, one of the interesting observations with that. If you look at the other data sets like Fashion MNIST, which is a black-white data set, and apparently you can see the performance degrades, but not as much. But the performance gap between the two is kind of interesting. You can see that the gap with the far right, for example, is much greater than the gap, for example, that you see on the far left, with different degradations. Here I use three different degradations. Gaussian noise, which means pixels are added a Gaussian random variable, a realization of a Gaussian random variable. And there's also a Gaussian blur, okay? And also the salt and pepper noise, okay? And I use different colors to characterize each one of these degradations. How about turbulence? Well, this is one of the things that IARPA was interested, because in many circumstances, machines are asked to recognize people at very high distance, but there is this, the atmospheric pressure changes, atmospheric temperature changes, which leads to a turbulent image that you can see here. The right without turbulence, left with turbulence. Unfortunately, it's a PDF. This was supposed to move to show you the difference between the turbulent image and without it. So you can see that the performance improvement with this modification in the architecture is apparent. I'm not going to go through the details, but what my proposal here is that by changing the architecture based on the human brain's separated processing of spatial frequencies could help you, particularly for these hard, challenging circumstances. Okay? And if you want to explain it through some heat maps, see if, where the networks are looking at when they're making such decisions, making classifications. We realized that the bionets, the biological plausible model, seem to focus on sometimes different regions, but when they look at the same place in the image, the focus is a bit more observable. In other words, if you zoom into these images, you will realize that the ResNet Bio 50 is showing a bit more focused heat maps compared to the heat maps of the ResNet 50, and here as well. So let me just show you here these two. So this is ResNet 50 original, and this is the biological inspired. As you can see, the biological inspired heat map has less regions colored with red and yellow. Okay, like this. Okay, so that's a good sign that we're extracting features that are probably more important than the rest of them. So if you just try different, as you can see, the high frequency components are kind of feed forward to the later residual cells. You can actually increase that. You can actually increase the depth of the network and see if that helps you. And it turns out that it helps, but not as much if you increase furthermore. For example, you've got 2, 3, 4, 5, then but the interpretation of these weights are kind of hard to make at this point. So what I did actually, I looked into, the model, and I trained the network. I look at these W's, you can see the W's, and write the X bar in terms of X, and this is what you get, okay? And you can also write the X double bar in terms of X bar, right? In this simple model, you can see the depth is 1, breadth is 2, depth is 2, sorry, 2 by 2. So, and this is a kind of the simplistic version of the proposed idea. And if you just replace these X bar with the expression above, for example, and you do all these algebraic calculations, then you end up something like this, okay, at the bottom of the picture. You can see that there's a main branch and also residual branch. The residual branch has a bunch of weights that you already learned, and the low-pass filtered weights and high-pass filtered weights, you can actually combine them somehow. But how? How are we going to combine them? Well, the idea is something like this. So what I did is I look at the low-pass filtered components' weights and multiply that with a Gaussian. And the Gaussian is scaled by the sigma, because those sigmas are also not learned, okay? Sigma is a parameter that tells you how much blur you want, okay? That's also a parameter that should be learned. So I scaled the Gaussian with that the belly of this, not the magnitude, but the belly of the Gaussian is modified by the learned sigmas for both high and low spatial frequencies, filters, low and high spatial frequency pass filters. And the magnitude of these sigmas are modulated by the weights, okay? The weights that you learn or do this, that you learn through training, okay? So these weights are the ones that are learned. And if you just multiply them and then draw these heat maps, you realize that, so there are 4 stages in the ResNet 50, and each stage I plotted these heat maps and show you that the high frequency is preferred in the first stage. In the second stage, there's a certain transition, and in the third stage, the low spatial frequencies are preferred, okay? So therefore, this is not something that we were expecting, right? So the high frequency spatial frequencies are prioritized in the processing for this model, okay? So there are more results here, so I'm gonna skip. This is also another result with CIFAR 100. So the previous result was for 10, and this was for CIFAR 100. And I'm also showing you with different depths, so depth 1, depth 2, by just feed forwarding these high spatial frequencies further down in the network. And it realized that, same kind of trend appears, apparently. The high spatial frequencies are prioritized compared to low spatial frequencies. Okay, finally, I'd like to show you some EEG results because this is an interesting result, which made us question that we know from previous examples, previous literature, that low spatial frequencies are prioritized, and then high spatial frequencies are seen to guide the low spatial frequencies. That's what we know from the literature. So we basically start questioning it because this network that I've shown you seemed to mimic the human network and showed us that the high spatial frequencies are prioritized. So then we would like to just run an EEG experiment, see if that's a true statement that we find in literature. Okay, here that we have shown multiple pictures to individual subjects, and some of them are clear images, some of them are blur images, some of them high spatial frequency images. Okay, the one, for example, see on the left, you can see it's a high spatial frequency, and the blur is a low spatial frequency, and that's a clear image. And there are also, the experiment goes as follows. You basically show 300 milliseconds an image to a human, and then hook up some electrodes on the brain. That's how the EEG works. And then you collect some multivariate data from the electrodes. And there are also some breaks, of course, because that could cause lots of habituation while the subjects are running an experiment. And we just wanted to see, it's a very simple post-processing, just let's see the signal that's due to high and low spatial frequencies. You know, when humans are exposed to high and low spatial frequencies, what happens to their brain signal? Okay, so it's an ERP called evoke, response that we would like to see the evoke response, because you show 300 milliseconds and then the image disappears. So therefore, that causes an evoke response in the brain. Okay. And there's also interstimuli time, and we show face and objects and blur, sharp, clear. So there are 6 different combinations. And there are sometimes repeats to make sure that the subjects are following along the experiment. And these are for attention checks. Let me jump to here. So here you can see that I actually encoded with different colors, low spatial frequency component, LSF, that's blue. The red is high spatial frequency images, that's red, and the green one is full resolution. And what I am drawing here is face plus object, so I don't really differentiate between face and object. I just averaged over all face and objects and all subjects. And I realized that, you can see on the right, perhaps I can show it to you. So here you can see up to 200 milliseconds, this red one seems to lead the blue one, right? The blue one's peak actually comes a bit later than the peak of the red. Okay? So you can see that red is high spatial frequency, not low spatial frequency. So therefore, up to 200, about 200, around 200, you see that the blue line now starts leading the red curve. Blue line is the LSF, the red line is the HSF. So therefore, what literature usually focuses on is the later stages of the processing, not the early stages, because early stages, high spatial frequency seem to lead the low spatial frequency. Whereas after 200 milliseconds, the low spatial frequencies start leading the high spatial frequency, which basically incongruent with the hypothesis that the high spatial frequency could be guiding the low spatial frequency. That's what we find in the literature. However, we see something different in the early stages of perception. Okay, that the high spatial frequency leading. So that makes us, and you can also kind of see the same thing for face, objects, if you actually divide this into 2, or you can look at the low spatial frequency only and high spatial frequency only. You can see that there's no difference between face and objects, but that difference is still there, like the early stages of the perception and later stages of the perception. And that is also in parallel what we found with the neural network design, right? Remember, in the neural network design, we also have seen something similar here, that the high spatial frequencies are processed earlier in the first stem cell of the ResNet, and then later the low spatial frequencies are more preferred. Okay, so that's the reason why we actually started the EEG experiment, to make sure that validate that the human brain does the same thing, although we're trying to make sure that the computational model has a similar architecture to the human brain. And then we see something different, in behavioral studies. Then we realize that perhaps we should look to EEG, see if there's alignment. And similar things can be observed. So what you're seeing here is just 3 different channels in the visual cortex, O1, OZ, O2. I'm sometimes skipping some of the details, so please interrupt me if you've got confusion. But here, only 3 channels are used, O1, OZ, and O2. But if you look at all the channels, like the topographic maps, you also kind of see a similar thing. You can see that the blurred space, which is low spatial frequency, you can see they're kind of almost the same, but mostly high spatial frequency is leading because it becomes red earlier than the low spatial frequency. But the high spatial frequency stays there longer. Okay, it stays on the frontal lobe, frontal cortex longer, and then it comes back to the visual cortex for final fusion. Okay, so apparently high spatial frequency is kind of prioritized in the early stages of the perception or processing, but then those spatial frequencies takes the lead. You can see that takes the lead, and then it actually arrives at the visual cortex earlier than the high spatial frequencies. Okay, so therefore these can now guide the high spatial frequency when the high spatial frequency comes back to the visual cortex. Okay, and you can also kind of see different connectivity. If you just look at the connectivity analysis, you will also see that there's also hemispheric differences. Right and left kind of play different roles. For example, here, I'll just jump to the conclusions. Here you can see that right hemisphere is the red line, and the blue line is the left hemisphere. This is for low spatial frequency. So the low spatial frequency seem to be processed by low left and right, okay, in the early stages. But then, after a certain point, there's no difference between the low and high. But the high spatial frequency seems to show something interesting. You can see that the left hemisphere seem to not involve in the processing of high spatial frequencies in the early perception, and then they develop. But then the right hemisphere seems to be the dominant hemisphere that processes the high spatial frequency. Okay, so there's a difference between left and right hemispheric brain regions. So we also want to run some extended tests on larger class data set just to make sure that these conclusions can be extendable to these data sets. Like, for example, ImageNet or EchoSet. As you might guess, these data sets a bit more, larger voluminous data sets. Therefore, it requires more computational power. EchoSet is a ecologically motivated image data set. And so here, indigenous behavioral similarity, feature map correlations with the baseline, which we have done earlier. We also have mathematical arguments. I don't have time to go through them, but what we found by just running a stability analysis, that we ensure by choosing the weights appropriately for the low spatial frequency components, but not high frequency, supporting the idea that low spatial frequency components. So if you look at the dynamic, there's some idea called interpolation of neural networks. Okay, so these neural networks are discrete architectures. In other words, you've got a certain number of layers, and there are a lot of work in the literature that makes this discrete layered network as a continuum. Okay, and that continuum basically helps you to analyze such a network using differential equations. Okay, so what we did we did the same thing. We formulated the neural network as a differential equation, and then see if the weights that modulate low spatial frequency and high spatial frequency, and weights that modulate the high spatial frequencies, what's their effect on the final stability of the dynamic system. And it turns out that the stability of the network is ensured by the low spatial frequency components, not the high spatial frequency components. So that also kind of helps us that low spatial frequency is the dominant factor, still the dominant factor in the continuum range. However, if you discretize the network, we realize that high spatial frequency may not be guiding, but it's prioritized in the early stages of perception. Okay, so these are the two different conclusions. And of course, use of hemispheric spatialization. We create 2 copies. So for example, how are we going to use the hemispheric spatialization? We can actually have 2 copies of the same network, and one for example, can actually process low spatial frequency specifically, but that would resemble to what left hemisphere does in the brain. and also another copy of the network that can actually resemble or mimic what the right hemisphere does in the brain. Okay, so this can also be done, using some of the results we already have gathered from our EEG experiment, like in STEM1 could be. This is just an idea. We haven't run that yet, but this is an idea. Both hemispheres, for example, prefer low spatial frequencies in the STEM1. Okay, and in STEM2, left hemisphere could actually prefer LSF, the low spatial frequency, and right hemisphere can actually prefer high spatial frequency. In STEM3, for example, left hemisphere can prefer high spatial frequency, and right hemisphere could prefer either, because we didn't see any difference between the two. And finally, the left hemisphere, the STEM4, could prefer high spatial frequency, and right hemisphere could prefer high spatial frequency. And you can actually have, just expand that network something like on the left-hand side and see if the performance improves. Okay. Anyway, let me come to the questions and conclusion. First and foremost, the idea behind this study was, from theoretical perspective, is to understand the interesting relationship between nativism and empiricism. In other words, what's the precise characterization of contributions of genetics, and which means pre-wiring, your architecture is already developing, while you're a little child, and the environmental factors to the human development. When you have a skill, for example, which is in many literature is the definition of learning, where does that come from? Is it coming from genetics that's already pre-wired in your brain, or it's coming from the environment in which you are raised by your parents? Okay, so that's kind of the nativism and empiricism, and I think those studies could be used, I believe, by changing the training mechanism, for example, making it more developmentally plausible, and then see, if such a plausible idea, for example, could be mimicking how human child, for example, develop such skills. Well, see if such an emergent behavior comes out from the neural network. So it's a long discussion. There's a huge debate in the literature about nativism and empiricism, and I think such models could help create some, reasonable answers to such a question. The second is training, stimulation and optimization mechanisms. These have significant impact on the architectural and structural modularities to develop. So once you set when you set up the architecture, then the question is, how are you going to train such a network? As I said, these are a couple of things. And one idea which seemed to be helping us is the modularity. How are you going to enforce modularity into your neural network? And one simple idea here is just to change the training mechanism. For example, make it more biological plausible, as you see on the bottom of the page. Why don't you just train your network in this particular direction, rather than just use, okay, here network, here's the bulk of the data, just use it, okay? Perhaps this kind of training can help, develop some modularity within the network. And biological mechanisms, they're unable to, create modularity. And once such modularity develops, then how does the training proceed, right? That's the next level question. And tools for measuring the degree of modularity. How are we going to measure the modularity? Well, maybe the most straightforward way is to use psychophysics, like some of the neuroimaging techniques, like fMRI, AEG, MEG, and maybe less so for computational models, because we don't really know. We can't really hook up some electrodes to the neural network model to create, or to extract some information. What you can use, perhaps, maybe a tool to use is information theory. You can use information theory to see how the entropy changes, how the mutual information changes, over time or over different layers of the network. And architectural neural formulations eventually lead to changes in perception and interpretation of external stimuli. We have an hypothesis. There's a continuous feedback process between the external conditions and innate mechanisms, which is encoded into the architecture, and that evolves with the internal biological substrate architecture, okay? So maybe you don't know, but perhaps in the first couple of weeks, most of the processing in the baby is a feed-forward. There is no recurrence. But later on in the development, you basically form recurrent connections, which are basically now taking the output to the input, okay, and then creates this whole dynamic network. So our networks are fixed. They are feed-forward or maybe recurrent, but they're fixed. they're not changing over time. But if you look at the human being brain or the child brain, you will actually have a lot of stuff to get the inspired of. And this study and maybe the following studies along this line are trying to mimic the humans in every way possible to make sure our networks could guide us in terms of brain studies as well, because brain is a really complex idea, very complex, architecture. Finally, before I quit, I want to show you some preliminary results with different data sets using biomimetic training. Biomimetic training is dish training, okay? And you can see that biomimetic training actually helps. And that's kind of a validation of this. The green data is with the biomimetic training, and the blue one is the ResNet 50 without the biomimetic training. I'm sorry, the biomimetic training is on the left. I was referring to the other slide. In this slide, you see biomimetic training on the left and no biomimetic training on the left, and biomimetic on the right. And I also show you different networks, ResNet 50 and ResNet Bio 50, the biological plausible model. See, which one is benefiting from what, okay? And you can also, in the next one, you see that I also put the ResNet Bio and ResNet 50 and ResNet Bio on the same plot using different degradations. And apparently, you can see, although you lose a little bit in the high performance regime, but as your noise level increases, in other words, the degradation increase, then the green ones, you see, seem to perform much better than, which is the biomimetic training, which much better than the, so here you see ResNet Bio 50, which is the architecture I proposed, as I talked about earlier. But the biomimetic training also is helping you. So that training mechanism still gives you a bit of a margin of improvement on the performance as well. Okay, I'd like to thank you for coming, and I hope I used my time efficiently. If you have questions, I'll be happy to. I'd like to answer.
We see some performance training mechanism.
Can you ask again?
I mean, we see like some improvements in this graph, but in terms of the computational complexity, can you comment on this?
So that's a good question. So there are two things. First one is the architectural change. In the architectural change, as you might know, that the ResNet has millions of parameters, and so let me just jump onto here. Okay. So the only parameters that I'm introducing is in this full cell is the low pass filter sigma. That's the Gaussian. Since it's a low pass filter, that has to be a parameter to change the cutoff frequency. Okay. So there's one parameter here, one parameter here. These are weights, okay, and extra weights here. Okay. So you actually add for a specific cell 6 more parameters. However, these filters have lots of parameters, and you have like a bunch of them. So the percentage increase in the percentage, in the number of parameters is less than 1%. So therefore, the complexity of training this network versus the ResNet 50 original is only like a little minor, so it's kind of negligible. It's not that big. But while you're running the training mechanism, like the optimization techniques I use, these are tested extensively for conventional schemes, not for this. So because of, I believe there is the convergence problem with this scheme. There are sometimes this scheme does not converge very well, so therefore it takes a bit more time because of the training. And I believe that the optimization cost function or optimization techniques like backprop, they could also be made biologically plausible to help with that convergence issue. Okay?
Thank you.
But there's that too. It's not just the parameters.
Thank you.
Yes?
First of all, if my question is not going to be relevant to presentation, but
Don't worry.
okay, let's see how it goes. I see. Okay. How does the low spatial frequency actually turn a blurred image or a blurred recognition into a meaningful recognition by guiding through high spatial frequency or the vice versa situation. And if it's a meaningful question, then how did you impose this to the training set of the machines, I would say?
So let me reiterate
Yeah.
what you have asked. Okay, see if I understood correctly.
Yes, sir.
So you're saying that how low spatial frequencies are guiding
Yes.
spatial frequencies.
Yes, sir.
So that's $1 million question.
All right.
I mean, even in the brain, for example, we don't know the exact answer how they're guiding, but we believe it guides because of the way that they're merging. You know, they're all separating and then merging again in the brain. And through extensive experiments in the literature, people realize that low spatial frequency could be guiding. They always use like could be guiding.
Could be.
It's not like, you know, exactly we got yet, because unfortunately, even if you have access to neuron level information in the brain, since it's so complex, in other words, the neuron level information doesn't tell you in the higher level of abstraction how does that, for example, manifest itself in the recognition. We don't know that stages in the brain. That's why learning or studying the neurons doesn't really entail say something about what exactly the merge is or how the recognition happens or perception happens. So that these results are usually based on, you know, or could be guiding, would be guiding, etc. So my understanding, but computational models could help. I mean, it can actually provide. That's why we run some mathematical analysis. See if the mathematical analysis could, you know, help us understand the phenomena. But we haven't come to that point yet because we're trying to make sure that that model with this different training mechanism is a good model of how brain develops and learns and create this percept of objects around us. So I'm sorry that I wouldn't be able to answer your question very well, but the low and high spatial frequency and their interaction is quite interesting. But the guidance, the precise definition of guidance is unfortunately absent. I think maybe in a very high level, I would say low spatial frequency, when it's processed, it creates these feature maps in the computational framework. And those feature maps could be, for example, misguiding you. Right? Some features, for example, were not relevant. Some of them are not really expressive enough, etc. But then the high spatial frequency feature maps could create such expressiveness in the later stages of the network before you do the classification. Then feature fusion, as we know from the literature, feature fusion has been, you know, something that is practiced over and over again by different people. That feature fusion seemed to work very well. And maybe a similar phenomena is taking place in the brain. I'm not sure.
Yes, sir. Thank you for being humble by answering the questions. But one small question I would say. Then how do we actually impose it, or how do we teach this? The way
Training.
To the training
The way
sets.
we train now is like with the biometric. We basically start the training the neural network using low spatial frequency images, basically. And so, you know, I think you probably have trained neural networks, right, in your past experience. So there's something called epochs, right? There are epochs. So, for example, if I run like 50 epochs, what I do based on the functional change, because there's a certain functional change in the acuity, color perception in the babies in the first 6 months, for example, or maybe take a year. And then look at these functional changes of all these color, for example. For example, red color maturizes earlier than the green and yellow and other colors, for instance. So there are all these details. So what you do with basically, you look at for example, in the first 10 epochs, you train with the black and white blur images, okay? And then maybe in the next 10, you start introducing red, and then maybe reduce the blur a little bit. And then in the next phase, maybe reduce the blur even more, and then introduce green and blue, for instance. You know what I mean? That kind of training to make it more, you know, resemble to how the human babies are trained.
See, sir. I admit that I am enlightened by your presentation. Thank you, sir.
Happy to hear that. Question?
Öncelikle çok teşekkürler bu güzel sunum için. Ben Türkçe soracağım. Bu öğrenmenin beynin modülerleştirmesi ya da modüler öğrenme dediğimiz bir kavramdan bahsettiniz. Ya bu şey anlamına mı geliyor? Hani beynimdeki belli sinir hücresi öbekleri belli bir iş için özelleşiyor. Yani diyelim ki kırmızıyı tanıma bir yerde odaklanıyor, yeşili tanıma bir yerde odaklanıyor.
Yani evet.
Bu şey sizin bu bahsettiğiniz kavram bu yapay sinir ağlarında ya bir nevi böyle ardışıl bağlı öbekler mi yoksa birbiriyle paralel çalışan öbeklerin oluşması mı tercih edilecek bir şey? Hani ve bir de bu öbekli öğrenmede şey gibi bir durum var mı? Önce diyelim ki siz kırmızıyı öğrettiniz. Yani oluştu. Bir takım hücreler, işte katsayılar. Ya bu arada çok yapay sinir ağlarına hakim değilim. Hani terminolojiyi yanlış kullanabilirim, kusura bakmayın. Bir yerde oluştu. Daha sonra şey gibi bir yaklaşım var mı? Hani biz bu tarafı bir kapatalım. Yeşili başka bir taraf öğrensin. Sonra bunlar işte çalışırken paralelde de birbirleriyle hani ardışıl bağlanmasında eş zamanlı çalışsın falan gibi. Bu şekilde bir öğretme yaklaşımı var mı? Ya da sıralı yaklaşımın bu tür bir etkisi oluyor mu? Yani siz şey dediniz önce düşük frekans banttaki şeyleri verip sonra yüksek frekanslara geçersek işte modülerizasyon, yani öbeklenme artıyor gibi bir şey anladım ben. Yani böyle bir mekanizmada yani zaman daha doğrusu sıralama yerine böyle hani bir takım grupları kapatıp açarak falan öğretme söz konusu olabilir. Özetle.
İyi bir. Ben de İngilizce mi cevap vereyim, İngilizce mi cevap vereyim?
Türkçe cevap verirsiniz.
Birincisi şöyle bir şey söyleyeyim. Şimdi birincisi o kadar low level şey olunca karıştırıyorum İngilizceyle maalesef.
Soğanları sorun değil.
Low level şeylerdeki modülerasyon şeyde gerçekleşmiyor. Beyinde gerçekleşmiyor. Yani mesela kırmızıyı özel işleyen bir bölüm, yeşili özel. Bu seviyede değil. Benim söylediğim seviye şu. Mesela frekanslarda oluyor. Mesela low and high spatial frequency'ler var ya, onları ayrı yerler.
Konumlar farklı onların yani. Kaskat bağlı değil mi onlar peki?
Çok aşağı seviyelere inmiyor. Yani mesela bileyim sesle görüntü mesela farklı yerde işleniyor gibi. Anlatabiliyor muyum? Yani belli bir seviye var, onu tanımlaması da zor. Ama yani mesela farklı renkler farklı yerlerde işlenmiyor mesela. Onu söyleyebilirim mesela. Önemli olan biraz perception'a dair şeylerin olması lazım. Mesela low and high spatial frequency perception. Mesela siz biraz önce gösterdim, high ve low spatial bir tane face gösterdim. Tanıdığınız bile olsa high spatial frequency, direkt sadece high spatial frequency görseniz mesela zorlanıyorsunuz bulmakta. Low spatial frequency'ye ihtiyaç duyuyorsunuz. Anlatabiliyor muyum? Mesela bu tanımakla alakalı. Olduğu için low and high için farklı hardware ayarlanmış beyinde. Ama mesela kırmızıyı, mesela aynı yüzü kırmızı görseniz ya da yeşil görseniz arasında çok farklı olmadığı için bu kadar.
Tamam, anladım. Peki bu düşük ve yüksek frekanslar ardışıl mı birbirine bağlı? Yani önce siz hani tanırken de bir şeyi böyle bir düşük filtreden geçiriyorsunuz, kabaca anlıyorsunuz, ondan sonra yükseklere yapıyorsunuz. Yani birbirinin ardına mı geliyor beyinde?
Yani beyinde öyle. Beyinde low spatial frequency önce işleniyor. Burası
o hızdan anlamıştınız galiba, değil mi? Hızdan anladınız onu.
Benim gösterdiğim
Kaskatlı.
çalışma var. Benzer şeyler söylüyorlar. Başka AI kullanıyorlar. İşte MEG kullanıyorlar. Başka invasive teknikler var. Makaklarda da gösterdim makakı. Benim değil o. Başkasından aldım. Yani nöron seviyesinde de sinyallere baktığınız zaman low spatial frequency önce işleniyor. Orası kesin. Sonra high spatial frequency işleniyor. Fakat paralel mi işleniyor? Paralel işleniyor. Yani low spatial işlenirken high spatial beklemiyor. O da işleniyor. Ama biraz daha
ayrı kanallardan, ama diğerinin işleyişi daha hızlı olduğu için biz hızlı farklı görüyoruz.
Low spatial frequency'nin kanalının donanımına bakarsanız göreceksiniz zaten. Myelination diye bir şey var. Nöronlarda myelination oluşmuş. Yani myelin olmuş. Myelin de hızlandırıyor transferi. Mesela parvoda myelination yok. Myelination olmadığı için ya da az oluyor. Dolayısıyla oradaki transfer daha yavaş ilerliyor. Yani o spike'lar, ben tabii ki şu anda hepsi continuous düşünüyoruz ama spike şeklinde gidiyorlar. Spike'ları birinden diğerine iletmek zaman alıyor. Zaman aldığı zaman high spatial frequency aslında işleniyor. Spike'ların formu değişiyor, rate'i değişiyor falan, bir işleme var. Fakat buluşmadan evvel low spatial frequency tamamen bitmiş oluyor zaten. Anlatabiliyor muyum? Ve başka işlemler yapıyor low spatial frequency. Yani mesela bazen sadece low spatial frequency kullanarak da perception yapabilirsiniz. Sonra high spatial ihtiyaç bile duymayabilirsiniz. Özellikle tanıdıklarda falan mesela low spatial frequency hızlı işleniyor ya, pat diye tanıyabiliyorsunuz o kişiyi. Onun nedeni şu: low spatial frequency'ye özel ihtimam gösterdiği için beyin gerçekleşebiliyor o tanıma işlemi. High spatial frequency ihtiyaç bile kalmıyor mesela.
Sonuçta varyans daha düşük yani low frekanslı. Değil mi yani bir nevi? Çok daha az ayrım var belki.
Hesaplamalı olarak da bir sürü neden getirebiliriz. Ben sadece beyin tarafında ne olduğunu söylüyorum. İkisi de işleniyor. Yani sizin sorunuzun cevabı o. Paralel işleme, beyin inanılmaz paralel. Yani öyle biri diğerine beklemiyor kesinlikle. Her şey paralel işliyor. Fakat hızlar farklı. Hızlar farklı olduğu zaten learning'in de parçası hızlar olduğu tahmin ediliyor. Yani hızlı öğrenme gibi böyle değişik değişik skill'lerimiz ya da ne bileyim mesela birçok öğreniyoruz mesela memori de öyle mesela. Memorinin gelişmesi özellikle çocukluk zamanında bu hız, iletim hızlarının farklılığından kaynaklanıyor. İletim hızlarının farklılığı learning'in olmazsa olmazı diyelim.
Yani siz de eğitim sıralamasını buna göre vererek bir fark yaratmaya çalışıyorsunuz.
Architecture'ı zorladım. Yani architecture'ı zorlamadığın zaman o modülerasyon mesela biomimetic training var ya bu işte çocuk nasıl şey yapıyor.
Evet.
Onu ben normal ResNet 50'ye verdiğim zaman modülerity çok iyi olmadı. O yüzden
Biyomimetikte neden olduğunu düşünüyorsunuz? Yapı uygun olmadığı için.
biomimetic training'i normal bir ResNet 50'ye verdiğim zaman istediğim modülarasyonu elde edemedim.
Çünkü o insan beyniyle belki daha az uyumsuz, değil mi?
İnsan beyninin mesela Poggio var işte MIT'den işte çalıştığımız. Mesela onun bazı makaleleri var. Orada şey diyor. Hani ya diyor işte insan beyni aslında ResNet 50 yakındır. Normalde ilk üreten kişiler biliyorsunuz. Beyinden inspire olarak üretmiş bir şey değil. O Poggio'nun ve öğrencilerinin söylediği şey şu. Ya diyorlar bunun iyi performa etmesinin nedeni aslında beyine yakın olması.
Ama yanıldıkları bir şey var o zaman belki.
Belki de
Bir şey var belki.
Çok böyle kesin sonuç yok hani ortada. Hani bir sürü data var, eksperiment var ve yorum yapıyorlar sonuçta yani bu eksperimentler. Yani olmayabilir. Yani beyine istedik şimdi şöyle bir beyine yakınlık var. Mesela onların bulduğu belki yakınlık benim yakınlığımdan daha az bir yakınlıktır mesela. Bilmiyoruz. Belki ben daha çok yaklaştırmış olabilirim. Tabii o da var tabii. Yani hangi beyin diyoruz ama hangi beyin? Hangi zamandan beyinden bahsediyoruz? Hangi space time'daki beyinden bahsediyoruz? Öyle söyleyeyim yani. Mesela biz Mars'a gittiğimiz zaman aynı beyine mi sahip olacağız? Yani ya da ne bileyim uzak bir Andromeda galaksisinde yaşamaya başlasak orada aynı beyine mi sahip olacağız? Yani bu sadece time değil, space time. Space time'daki beyin. Başka soru var mı? Bu da ben DSS'e katılmıştım Florida'ya geçen sene. Orada bir poster sunmuştum da oradan bu da gün batımı arkadaşlar. Onu da söyleyeyim dedim yani. Eksik kalmasın. Peki teşekkür ederim. Sağ olun geldiğiniz için. Umarım faydalı olmuştur.